Good afternoon, everyone, and welcome to the Replace Educational webinars. For those of you who don't know the Replace project, the aim of this project is to collect the expertise we have on alternative methods to animal testing in one central open access database. In the database, each method is associated to the name of a scientist because scientists submit the methods themselves in the database. So if you are working in Belgium on alternative methods, please feel free to add your methodology in the Replace database or contact us. And one of the other goals of the project is to share the knowledge about alternative methods. And for this reason, we have launched this series of educational webinars to promote the expertise we have at the moment in the Replace database. So in the webinar session of today, we are going to listen to the presentation of two speakers from the database from the University of Hasselt and the KU Leuven. But before we actually start, I would like to give you some housekeeping rules for the webinars. So today we will have two speakers, as I said before, and the Q&A sessions will be at the end of each presentation. So if you have any questions in the meantime that you would like to ask to the speakers, please don't hesitate to add them in the chat. Also, this webinar is accredited by the Flemish and Brussels regions. So if you would like to receive a certificate of attendance, don't hesitate to add your name and email address in the chat, and I will provide the certificates in the coming week. And finally, this webinar is also recorded, so it will be placed later on the Replace YouTube channel so that you are able to see the recording in open access. But now we are going to dive into the presentation. So the first presentation of today will be given by Dr. Bella Manshian. So Bella is a team leader at the Alternative Translational Models Group within the Department of Imaging and Pathology at the KU Leuven. And currently, she's leading the group on drug delivery system and 3D ex vivo precision cut tissue slice technology on chip. Her main interest lies in studying toxicity of environmental agents, chemicals, and nanoparticles, and also developing 3D ex vivo translational models from human and mouse organ tissues, including lung, liver, ovarian cancer, Crohn's disease, cystic fibrosis, and brain. So Bella, the floor is yours.
Thank you. I will share my presentation first.
Yes.
I think it will be this one. Oh, that's, I'll try and see which one it is.
Yeah.
Can you see the slide? Is it in the presentation mode?
Yes, it's perfect.
Okay. So just let me fix this as well. Okay, good afternoon, everyone, and thank you for being here. I would like to start my presentation today by stressing the importance of working towards non-animal methods. As you probably know, between the 1950s and 2010, the use of animals in preclinical research increased exponentially, and in response to the growing concerns of Dr. Russell and Birch introduced the 3Rs principles of replacement, reduction, and refinement to minimize animal use in preclinical experimentation. In 2010, the European Union joined in and published a directive aimed at advancing the implementation of the 3Rs. And in 2022, the FDA announced that animal testing is no longer required before human drug trials, which was excellent for this initiative. And another noble initiative for supporting these efforts is where we are today, the creation of the Replace database in 2017, which centralized non-animal methods to further reduce and replace animal testing in Belgium. According to statistics from the European Union, animal use in 2020 was just under 8.5 million, which remains high, of course, yet marked a significant decrease compared to the two preceding years, indicating progress in this direction. These advancements have also driven the growth of the global market for non-animal alternatives, which is projected to reach 3.63 billion by 2028, which is good news for us. In the era of personalized medicine, there have been accelerated efforts to develop clinically relevant platforms to test drug sensitivities and efficacies and toxicity, etc. However, current screening and drug development models often lack robust clinical translatability. This leads to prolonged research timelines of 12 to 15 years and billions of euros in investment, with only a small fraction of these discoveries, as you can see in this figure, from 10,000 discoveries down to one that is marketable. The primary challenge, according to me, does not typically lie in the efficacy of the drugs that are being developed, but in the limitations of the research tools that we're using, highlighting the need for more translatable models. Therefore, my lab has focused on optimizing 3D models with enhanced biological complexity and throughput, with the aim of advancing biomedical research. Our mission is to develop human representative translational models that would facilitate the progress of personalized medicine. To ensure this, our models need to possess sufficient complexity to reflect the individual scenarios. They need to be miniaturized for clinical applicability. They have to be cost effective and adhere to the principles of the 3 Rs and the NAMs and be compatible with high throughput applications. For this purpose, we employed precision cut tissue slices technology, which these tissue slices are sophisticated experimental platforms. They preserve the complex architecture and the cellular composition of human or animal tissues. These models offer a more accurate representation of disease pathology that also facilitates the in-depth study of disease mechanisms, drug responses, and specific effects within a controlled environment. The use of precision cut tissue slices also ensures uniformity because we are able to control what size we slice these slices. We can control their diameter, we can control their thickness. They also are, they have higher reproducibility and they give reliable data, making them particularly suited for applications in drug discovery, toxicology testing, and personalized medicine research. By maintaining the native tissue microenvironment, these models are able to simulate chronic diseases or cancers and other conditions like in viral infections, etc., thereby providing valuable insights into disease progression and optimizing the treatment strategies. The primary objective of our study was to establish a longitudinal culture of stable precision-cut lung slices. As you can see in our experiments, we successfully cultured these tissues for up to 14 days, maintaining the integrity of their structure and morphology. As you can see, the slices from the tissues that were prepared from before slicing the lungs, and then after slicing for up to 14 days. Analysis of key tissue parameters, including the area, perimeter, and the radius, revealed no significant changes over the 14-day culture period. Additionally, through the use of a multiplex immunohistochemistry system, we confirmed that the cellular composition in these tissues remained stable throughout the duration of the culture. When looking close up, we can see that these tissue slices retain the architectural integrity of the original tissue from which they are derived. Therefore, it is not necessary anymore to create these architectural structures because they are preserved. As demonstrated in these images, the lung slices preserve key features, including the alveoli, bronchi, and blood vessels.
I'm sorry, Bella, to interrupt. Apparently, some people are not able to see the slides, but from my side, I don't have any problem. But can you try stopping and resharing? Maybe
Okay.
it might help. And for the people who are not able to see the slides, maybe you can try to download the WebEx app, or you can also try to close it and reopen. I hope it can help.
Okay, just let me see. So stop sharing and then I share again.
You can share it again, yeah.
Okay, good. All right, so I think I was in here. So let's share again. So I cannot see myself now what is being shared and how. Just, sorry.
No problem.
I'm going to stop sharing one more time.
Yeah.
I'm going to try it again. Share. Okay. What are you seeing now?
Now we see nothing. It looks like the sharing has stopped. Okay. Can you confirm in the chat if you can see the screens? For me, it's okay, but okay, perfect. Okay.
Yeah.
It looks okay. Yeah. Sorry for interrupting.
No worries, no worries.
You can continue.
So we will continue from here. So because we wanted to understand better how stable these tissues are during the in vitro culture and to see metabolically if there are any changes in their status, because viability we thought was not sufficient information to know how the health of these tissue slices, we assessed some biomedical markers and observed some alterations in lactic acid production, as well as some fluctuations in glutamine, alanine, and proline. But interestingly, these changes were happening on days 2, 4, and 10 of these assessments. So in an effort to explore these changes, we initially supplemented the tissues with arginine. However, this intervention did not result in any significant impact on the tissue viability. And then we further optimized the culture of these slices, and we were able to circumvent this problem by supplementing the media, the tissues with media every 2 days, and this improved the stability of the culture. Now, you know that viability screening is an important assay, and in precision cut tissue slices as well, it's a crucial assay commonly employed to assess the health and the functionality of these tissues. And to ensure that the most accurate evaluation of tissue viability is being used, we conducted a series of tests. These included measurements of intercellular and extracellular ATP levels, a LDH assay, and the Alamar Blue HS assay. All three methods, as you can see, provided reliable and reproducible results. We could see that the tissues are remaining healthy in culture and that over time there's a slight increase in the viability, which is to be expected that some of these cells would be replicating. However, the Alamar Blue assay proved to be the most effective for us because it could longitudinally monitor tissue viability and the metabolic health in the same tissue slice over extended period. Therefore, we decided to continue all future experiments using this assay. And through the optimization of various experimental parameters, we were able to sustain tissue viability further up to 39 days. Our second objective was to develop a disease predictive model. Obviously, that is the goal of this whole these efforts. And to achieve this, we employed a mouse lung cancer model by generating orthotopic lung tumors. The lungs were subsequently isolated and processed using the Crumdic slicer, and tissue viability was monitored for up to 12 days, and we could see that the tissues remained viable. Throughout this time, the tissues preserved the morphology before their slicing and culture characteristics. And then we performed gene expression studies, which remained stable and were comparable to those observed in freshly sacrificed mouse lungs. And importantly, when we did immunohistochemistry analysis, we could see the presence of tissue-resident immune cells. Macrophages, neutrophils, and cytotoxic T cells were intact in these tissue slices, and notably more in the cancer tissues, where we could see that there was increased expression of activated T cells. And this would also confirms the ability of the precision-cut tissue slices to capture the complexity of the tumor microenvironment and its role in understanding immune cells and their treatment strategies. To validate the precision-cut lung tissue slices that was done based on the systems that we followed, but now we wanted to know whether these tissue slices are actually replicating the in vivo scenario. So we created another lung cancer model in vivo in animals, and we administered paclitaxel to the mice with lung cancer. At the same time, we prepared precision-cut tissue slices from the lungs of untreated mice with lung cancers that were not treated. And the slices were exposed to paclitaxel ex vivo at the same strategy as they were in vivo. Our results revealed that the ex vivo response of these tissue slices actually to paclitaxel closely mirrored the tumor regression profile that we observed in vivo, therefore confirming the suitability of precision-cut lung slices as a reliable model for studying therapeutic responses. We next explored the potential of precision-cut tissue slices for high-throughput screening applications. So we used luciferase-expressing cells and generated orthotopic lung cancer model again, and then we took these animals, the lungs of these animals, and then we created precision-cut slices from them. We looked at flux values in the living animals and ex vivo in the isolated lungs and in the tissue slices. Then we treated explored the viability of these tissue slices using the flux values that are from the cells that had carried the bioluminescence vector and doing Almar Blue as viability assay. We could see that the result obtained used with Almar Blue and the flux values were comparable, and this confirmed that the PCTS actually can be used in high throughput screening in applications where you can use bioluminescent or fluorescent reporters. We therefore, after evaluating the ability of precision-cut tissue slices to mimic the in vivo scenario of treatment with drugs like paclitaxel, we were curious to know whether these slices will also be functioning well in the context of nanoparticle treatment, because as you can imagine, nanoparticles are not the same as drug. Their uptake and their transport in tissues and in cells is different than a drug or a chemical. So in a recent publication, we had examined the potential of silver nanoparticles as adjuvants in immunotherapy, and we had shown that there is actually potential for reducing tumors when used in combination. To build on this, we compared these findings from that study with experiments that we performed on precision-cut lung slices from the cancer's lungs. The results from the PCTS treatment, as you can see here, closely mirrored those of the in vivo studies. This further validated to us that the PCTS is an effective and a reliable model for predicting both therapeutic and exposure responses, also in the field of nanomedicine and nanotoxicology. An immunohistochemical analysis of the precision-cut tissue slices demonstrated enhanced cell death and the greater presence of cytotoxic T cells and macrophages in the slices that were treated with the combination therapy compared to the control slices, which is also in line with the in vivo results. Then we wanted to see whether the PCTS can be actually functional in predicting other disease models. And one of the disease models that we work with is the precision-cut patient ovarian cancer slices. In this study, we assessed the potential the therapeutic potential of carboplatin as a chemotherapeutic agent and hormone replacement therapy using ovarian cancer patient samples. In the first case, carboplatin treatment of tissue slices from patient 7 did not show potential therapeutic response in our tissue slices. Upon comparing these results with the patient's clinical data, it was evident that the carboplatin chemotherapy had been ineffective in treating this patient, leading to the return of the tumor, and the patient was scheduled for a secondary debulking procedure. In patient 26, the clinical tumor pathology showed high estrogen expression, suggesting that this patient could benefit from hormone therapy. However, our results from the precision-cut ovarian slices from this patient showed otherwise, and our test suggested that the tumor might respond more effectively to chemotherapy with carboplatin. Upon reviewing the corresponding, I don't know what happened. Can you still see my presentation?
No, we cannot see it.
Can you see it now?
Yeah, now it's okay.
Okay. So, I don't know what happened. This is fine. So upon reviewing the corresponding clinical data, we found that the patient had indeed initially been treated with hormone therapy, but the tumor recurred and the patient was subsequently scheduled for chemotherapy. I also wanted to mention here that we further explored the use of bee venom, which is a therapeutic agent that we are trying to develop in my lab, to see whether they would be effective in treating the ovarian cancer in these patient samples. And as you can see in the gray line, the bee venom actually was showing some reduction in the viability of the cancer tissue, which if compared to carboplatin shows potential if obviously optimization of the treatment concentration and regimen is done. We also collaborate with the target group at KU Leuven. Here we utilize intestinal tissue from patients with Crohn's disease, and we prepare precision-cut intestinal slices. These tissues were kept viable for up to 15 days under our culture conditions. And here we examined the effects of two pharmacological agents, hermine and Janus kinase inhibitors, JAK, which are known to be clinically used therapies or drugs for Crohn's disease. And as anticipated, our findings indicated that the tissues treated with hermine exhibited improved health, with cytokine expression levels beginning to normalize towards control values after 24 hours of treatment, which was not the case with the JAK treatments. We also are able to culture liver slices, and here we assess the ability of the liver slices to retain their metabolic function. To investigate this, we treated the liver slices with the prodrug irinotecan. Now we know that irinotecan will only exert its toxic effect after it is converted to its active metabolite SN38. We compared the effects of irinotecan treatment with those of direct SN38 treatment, and in both conditions we observed a loss of tissue viability after 72 hours of treatment, which confirms that these liver slices retain their metabolic function as well, and they're able to break down drugs into their metabolic states, their metabolites. And as a part of a collaboration with Cienzano, we utilized these liver tissue slices to screen two mycotoxins, ziralenone and alternariol. As we know, these mycotoxins are very toxic, and because of their toxicity, in vivo experiments are challenging to conduct even in animals, and testing is typically carried out in vitro or using zebrafish models. But we were able to test these in the liver slices. Our results were consistent with those obtained from the zebrafish studies in the right lower corner, where toxic effects were observed at concentration of 10 micromolar and above, and where toxicity was higher after 48 hours. These results were also confirming the ability of these tissue slices to predict toxic effects of mycotoxin. We're currently performing additional investigations to see what are the underlying mechanisms of these toxicities. We've recently also initiated investigations into human COPD lung slices to study fibrotic mechanisms. Now, obviously, we have done some work on animals, but the aim is not to continue with animals. The aim is to continue with human tissues as much as possible. We successfully maintained these tissues in optimal culture conditions for 14 consecutive days. And when we tried to do characterization of the epithelial and endothelial cell populations using image-based flow cytometry, we could see that actually we are able to capture those characteristics of these tissues. And currently we're in the process of characterizing additional cell populations within these lung slices. Our third objective is to establish biobanks of tissue slices derived from both animal and human tissue sources. Now, you might think, why? Well, this approach would enable the generation of hundreds of tissue slices from a single mouse. That would be used for screening across various organs. Furthermore, biobanking will also be crucial in preserving valuable patient tissue samples for future testing. To achieve this, we stored the tissue slices using 3 different preservation methods: conventional freezing, controlled rate freezing, and snap freezing. Upon thawing these slices after having been frozen and placing them back into culture, slices frozen using the controlled rate freezing and snap freezing failed to regain viability that was comparable to the values prior to freezing. However, with the conventional freezing method, tissues regained their original viability, albeit after 2 days of being in culture, and that could be something related to it requiring some time to regain metabolic activity. Therefore, the conventional freezing method was the most effective for preserving and reculturing the tissue slices, and now we are preserving some of the animal slices that we're generating. During our optimization and as an ongoing work, we also monitor oxygen consumption in these tissue slices using 2 distinct systems, the Seahorse Analyzer from Agilent and the Recipher system from Lucid Scientific. Both systems enable the measurement of oxygen consumption in each well containing each of the tissue slices. A key distinction between these two is that Seahorse system performs endpoint assays, while the Recipher allows for continuous real-time oxygen monitoring over extended periods, including several weeks of culture. So measurements here were taken every 30 minutes under various experimental conditions. We varied the culture media volume, the serum content, the well depth, etc. The data shows that in these graphs shows that oxygen diffusion within the wells remain consistent. Those slight elevations that you see or the dips that you see in the second graph, these are related to the fact that at this time point the media was being changed and therefore the sensor was being detached from the wells and then reconnected. This was very good news. However, a notable difference in oxygen consumption was observed, which correlates with the type of media used and its volume. Additionally, the graph here shows that how oxygen concentration varies with different well heights, and this makes us arrive at the conclusion that conventional tissue culture systems can be useful, but could sometimes lead to oxygen levels dropping and being insufficient for these tissue slices, leading for these slices to grow in slightly hypoxic environments rather than being in normoxia that they should be in. We have circumvented part of this issue by using breathable membranes that we cover the plates with during all their incubation period. But a further step we took in this direction is our work in developing vascularized tissue slices on chip technology. So here I would like to let you think a bit further and consider the potential for eliminating or reducing animal testing while simultaneously improving accuracy of patient outcome predictions. A world where the biomarker you discover or the therapy you create is not just a breakthrough, it's guaranteed to reach the market. That is the dream that we want to pursue. And as you know, in recent years, there have been various methodologies that have emerged, and some of the most successful being organoids or organ-on-chips. And these are all have been quite successful. However, an ideal system would be a diagnostic companion that provides scientists and clinicians with insights into exposures, and they are sensitive, therefore improve outcomes while minimizing unnecessary toxicities and costs. So at the moment, we don't have such a platform that exists for clinical or research use. However, the horizon is not far, and we believe that in the coming decades or years, hopefully we should say, that these advances will become more realistically available. If you look at this table, you can see that amongst these, precision cut tissue slices actually stands out for its complexity. However, it struggles when it comes to throughput delivery. To address this gap, we aim to enhance this technology by implementing vascularized tissue slices on small, cost-effective chips that are easy to produce and reliable, ultimately improving the current limitations of the system. We have successfully developed various microfluidic chip devices and liquid interface systems. I will not delve deeper into this topic at the moment, but perhaps in the future I can rejoin and then discuss this further. Our culture studies have shown that tissue slices remain healthy and retain their structure within these systems. Currently, we are optimizing the vascularized chips to more accurately replicate the circulatory system, because the next phase of our work will involve culturing tissue slices from 2 or 3 different organs within the same chip and monitoring the interactions between these tissue slices. In closing, I would like to express my sincere gratitude to the Nano Health and Optical Imaging Group for fostering an environment in which my team can thrive. To my inspiring collaborators and to the funding organizations, I also like to thank the organizers of this webinar, Dr. Mode Everard and Dr. Mieke van Mulders, and all of the Replace team. And I appreciate all of you for being here and for your attention. And I will be happy to take any of your questions.
Okay, thank you very much for your nice presentation. I already have one question for you. So you mentioned different type of organs that can be used as a precision tissue cut slice. But I was wondering if it was applicable to all the organs, or and if not, what would be what would be the condition for the organs to be able to be adapted in using these methods? Is it possible that there is an organ that we say, oh, this one will never be can never be used as an ex vivo model?
Okay. Actually, we have been working with many different types of organ from animals. And at the moment, we have succeeded in culturing brain, pancreas, kidneys, liver, lungs, heart. I think these are them. Human tissues are much easier to culture, and they are less demanding for optimization. One thing, however, I would like to mention is that each different organ will require its own optimization, starting from the media conditions into the slicing protocol, into the monitoring the wells, because some tissues tend to settle down at the bottom of plates, some tissues tend to float. All of these differences will have to be accounted to have a successful model.
Okay, thank you very much. I see two other questions in the chat. So the first one is, what are the liver slice culture condition? So for example, media supplements or percentage of oxygen?
I didn't understand the last part of the question.
What are the liver slice?
What are media supplements and oxygen?
For example.
We do not provide our tissues with extra oxygen, so they are living based on the atmospheric oxygen levels. However, as I mentioned towards the end of my presentation, we have reverted to using breathable membranes to cover our plates to allow for better oxygenation and air exchange, and that helps their growth. And at the moment, we are busy with our tissue slices on chip technology, where we actually have oxygenation system which monitors and controls it better. With culture media, I guess if that person is interested, we could discuss the details of these things separately. But we have indeed different types of culture media that we use with the different tissue slices that we don't disclose of unless if we have to, because obviously this is a part of the optimization that we are performing.
Okay. And the second question is from the same person. Are special additives added to the culture medium after thawing the slice to help them recover?
No, but it is something that is worth considering. That's a nice suggestion. Perhaps by doing that, we can avoid the 2 days when we have to wait for the tissues to regain metabolic function. However, at the moment, we have done nothing. We just cultured them the same way we were culturing them before freezing.
Okay. There are 3 other questions in the chat. So the first one, what procedure do you recommend to measure LDH content of the tissue? So in brackets, to compare with LDH release in culture media.
Yeah, so we actually look at levels inside the tissues and in the media. So we just use the LDH kit. We did, if I go back, perhaps, I don't know how easy it will be to go back in the presentation. So we also had the controls. So I don't know how well you are able to see this. We also measured the intracellular LDH level at the different time points and the extracellular LDH level, and but we would not have done the intracellular ones because obviously we would have to lyse these tissues, these cells, to be able to acquire it. But both of them were kind of good in showing the health of the tissues. Partly, perhaps in our case, we did not have much issues because we had performed quite a lot of optimization. So our tissues were behaving in a good way, were growing in a good way. Perhaps if you have tissues where you are struggling with keeping them alive in culture, then other considerations have to be taken into account. If that's the case, I would suggest for the person asking the question to resort to using ATP measurements because they are more reliable than LDH measurements in that case. I'm not sure if I answered the question.
And there is another question. Could you further explain the use of permeable membrane in culture plates to tackle oxygen consumption variability?
So the oxygen consumption variability had nothing to do with the membranes. So we actually measured them in just normal plates. And in this case, we can't even put membranes because we have the device, which is a sensor, and it sits on top of the plate as a just like a lid, and it comes with the probes, and that is doing the measurements. What I was trying to say is that we do see that indeed there is a bit of low oxygen levels inside these tissues, which could also explain some of the lactic acid production that we were seeing initially. But when we put the breathable membranes, the breathe easy membranes, these have some pores that allow for oxygen exchange to be a bit better. And then we could see that we're able to culture these tissue slices for longer periods without observing any damage or any changes.
Okay. And the last question is, why did you add DMSO before freezing?
We just wanted to follow the same procedure we do for freezing cells. So in our lab, we use DMSO with FPS, and we wanted to replicate what we did with cells.
Okay. Thank you very much for your interesting presentation. Now we are going to move on to the second presentation of today, which is the presentation of Nathalie. So Nathalie Dirkx is a PhD student working at the FIRST lab led by Professor Esther Wolfs at Hasselt University. And in her project, she uses an alternative stem cell sources from wisdom teeth to model a peripheral neuropathy called Charcot-Marie-Tooth disease. And we are happy to discover more about her work right now.
Thank you for the introduction. I will share my screen first. Okay. All right, you're seeing my screen, right?
Yes,
just to be sure.
I can see your screen.
Perfect, thank you. Okay, so I indeed will be talking about dental pulp stem cells, and some of you might already be wondering, these are actual cells that we derive from human teeth, because in fact, human teeth are a very rich source of stem cells. And if we look at development, tooth development throughout life, we are not born with teeth, they have to erupt first. Once they do erupt, we have our primary deciduous teeth that we lose during childhood to finally develop our permanent teeth. And as you can see here, sorry, I think this is in the way. As you can see here, you see that there are several different stem cell populations present throughout these different phases, with even in our adult permanent teeth being present of within the dental pulp and as well as the periodontal ligament. Now, why is it interesting to look at stem cells in adult teeth? Well, God, sorry, I have some issues with my slides. Yeah, sorry, a slide skipped. However, what I wanted to say is that we have a specific type of adult stem cell teeth, that are often extracted, namely wisdom teeth, because in up to 70% of young adults, they cause a lot of complications, pain, even dental disease, and therefore they often have to be extracted. But therefore, teeth and adult wisdom teeth are actually very accessible, and they can easily be recovered from medical waste material as an ethical stem cell source. I'm afraid my slides are not moving. I think my internet is a little bit bad, sorry for that. In my research project, we are focusing mostly on dental pulp stem cells, or DPSCs, and these DPSCs can be found within the inner core or the dental pulp of these wisdom teeth. As you can see, this area is also highly vascularized, innervated by nerves as well. And the DPSCs that we collect from them are mesenchymal stem cells with very excellent cryopreservation and proliferation capacities, which makes them very suitable for biobanking. On top of that, they can also migrate, they have paracrine effects, which are anti-inflammatory, and they can differentiate into multiple lineages. On top of all of that, they are actually also very easy to handle and even to isolate. Now, you might be wondering, how do you isolate stem cells from wisdom teeth? Well, we do it using the explant method. And as you can see here, we actually mechanically fracture the teeth open, so we split it in two to be able to isolate the pulp tissue. Once we have it, we fragment it into little explants, which are then simply put into culture and left undisturbed. And then within 2 weeks, we see that the DPSCs, thanks to their migration capacities, will actually grow out by themselves. And here you can see an image of the exact moment at which this is happening. So you can see all these dental pulp stem cells migrating out. And as soon as they do, they soon start to proliferate very well. Now, similar as other mesenchymal stem cells, DPSCs can differentiate to the three lineage, mesodermal lineages, so adipogenic, chondrogenic, and osteogenic. But there is something more interesting going on as well. If we look at just DPSCs without any differentiation, we see that they already express basal expression markers of the neural and glial cells, such as beta-3 tubulin, neurofilament, and also the P75 neurotrophic receptor. By now, while most of the initial research focused mainly on DPSC differentiation towards dental ontoblasts, by now you can see that a lot of labs are working on a lot of different cell types derived from DPSCs, including hepatocytes, myocytes, even cardiomyocytes, pancreatic cells, and endothelial cells. In our lab, we focus on the use of DPSCs to generate neurons, but also Schwann cells. And why are these Schwann cells so interesting? Well, it is because there are a lot of limitations in current Schwann cell models. If we look at animal models, we can isolate primary Schwann cells, but the animal peripheral nervous system, of course, differs from humans. And it has been shown that for several peripheral neuropathies, such as Charcot-Marie-Tooth, that pre-candidate drugs that do very well in animal models fail to translate to human trials. So it is really clear that we need human models, or at least more clinically relevant models as well. On top of that, the isolation of primary Schwann cell yields a very low number of cells that are often overgrown by fibroblasts. So there are also practical limitations. On top of that, Schwann cells in their in vivo environment depend strongly on neurons and their extracellular environment. So when you isolate them from an in vivo situation, they actually undergo de-differentiation from their most mature myelinating state, so they are not that mature anymore. The same thing is true when we would isolate primary Schwann cells from humans. However, this isolation would mean that we would have to isolate them from actual human nerves, which can cause a lot of complications. So therefore, this is unethical, of course. For completeness, I will also mention the immortalized or schwannoma-derived Schwann cell lines. These are interesting to study biological processes, but it has been proven that these different lines vary a lot in phenotype of the cells, and especially in myelin and myelin composition. In fact, there are some schwannoma cell lines that do not even express peripheral myelin protein 22, which is the major component of the peripheral myelin sheet. Finally, like in many research fields, people are also often using iPSC-derived Schwann cells. However, because we already know Schwann cells have are differentiating and not in the normal environment, so these iPSC-derived cells really suffer from maturity issues, hence they are actually more Schwann cell precursor state. On top of that, protocols are also quite long, expensive, and often lack reproducibility among different labs. Now, why do we suggest our dental pulp stem cells as an alternative? Well, interestingly, our DPSCs are already derived from the neural crest lineage, which is also the lineage from which Schwann cell precursors and eventually myelinating Schwann cells are derived. So in contrast to iPSCs, for example, where we first have to do a differentiation to the neural crest, DPSCs are already a step ahead and can be more easily differentiated to myelinating Schwann cells. And indeed, our lab has pioneered a protocol to do so in only 18 days. And you can see here in these images that the mesenchymal fibroblast-like DPSCs become very nice bipolar Schwann cells with a very clear morphology. Of course, we also validated the phenotype, and we shown in different donors that there is significant increases in some well-established Schwann cell markers at the gene and the protein level, looking at, for example, the neurotrophic P75 receptor as 100, but also more mature markers that are more involved in myelination, such as myelin protein 0 and the extracellular matrix protein laminin. So based on this validated protocol to generate DPSC-derived Schwann cells, or DPSC assays in short, we are now working on a patient in a dish model for Charcot-Marie-Tooth disease, or CMT. Now, most people have never heard of CMT. However, it is the most common hereditary peripheral neuropathy worldwide. The disease is progressive, and at the moment, there is not a single therapy available for patients. Symptoms include motor and sensory issues, and often also affect the muscles. So patients often develop skeletal deformities within their hands, their feet, muscle atrophy, weakness, and this all results into difficulties with walking, with coordination. So daily activities can become very difficult. On top of that, patients also suffer a lot from pain, insensitivity as well, and overall fatigue. While CMT is a quite broad group of several different disorders that all result in the same phenotype, the most of the patients are affected by CMT type 1A. CMT type 1A, or CMT1A in short, is caused by a duplication of the peripheral myelin protein 22, or PMP22 gene, which mainly affects Schwann cells, causing them to become dysfunctional and dysmyelinating, which causes axonal loss. While we do really know that PMP22 causes the disease, and we have known for a very long time, still a lot is unknown about the actual normal role of PMP22. We do know that it is located in the myelin sheath, where it functions in maintenance, stability, as well as in Schwann cell functioning. However, a lot remains to be unraveled, and it is mainly due to the fact that there really is a lack of clinically relevant models for CMT1A, similar as there is a lack of these models for Schwann cells in general. Therefore, we are working on our project to use our DPSC assays to mimic CMT1A, and we started doing so by first overexpressing PMP22 ourselves. And you can see we have been able to do so using lentiviral transduction. You can see a very stable, strong increase in PMP22 in the DPSCs. But we're also optimizing a model in which we use CRISPR-Cas, because this gives us a more controlled and precise method to overexpress the gene. And on top of that is also an inducible vector that gives us temporal control so that we can overexpress it whenever we want during or after the differentiation. And while we are looking further into the effects of overexpressing PMP22 ourselves, we also thought it would be interesting to actually isolate DPSCs from teeth of actual CMT patients. Of course, it is not that easy to find patients that still have their wisdom teeth and that also still have to have them extracted. So we started an international search, and we did so with the help of an American patient organization that also funded us. And soon we already realized that, of course, you would have to adjust our workflow, because up to now, whenever people were using stem cells from teeth, they would go to the hospital with the right medium to collect the teeth immediately during the extraction, and then immediately isolate the stem cells in the cell culture lab. However, our patients are not always close to us, sometimes not even in the same country. So we optimized our medium so that we could safely send it to patients or even the hospitals if they want to collaborate in advance of the extraction, so that they can take it along. The teeth are still put into the medium immediately after the extraction, and then the tubes with teeth are sent back to us as soon as possible. And by optimizing this method, we are now able to still isolate these dental pulp stem cells up to 2 weeks after the extraction of the wisdom tooth. And by doing so, we have been able to collect a biobank of patient-derived CMT, patient-derived DPSCs. And as you can see here, we have a large patient population in Belgium, but also in the US. with these DPSCs, we are of course now differentiating them as well into our DPSC Schwann cells. And you can see that they also form these nice Schwann cell morphologies. And they also, so we confirmed this, that they also overexpress BMP22 before differentiating them, but also still after the differentiation. We also checked BMP22 at the protein level, and here we also see the increases that we expected. So now that we have our protocol to get our DPSC assays, we have a large library of healthy donors, but also of CMT donors with the BMP22 duplication, but also other types that could be interesting in the future. And we are validating and characterizing all these cells. But like I said before, Schwann cells depend a lot on their environment. So we eventually will have to go into co-cultures to be able to study demyelination. And interestingly, in the past, so back in 2014, a group was already able to show that healthy DPSC assays are able to myelinate sensory neurons from rats. And it is now our goal within my project to replace all the animal cells and really make a human co-culture model with increased clinical relevance. And therefore, we are using iPSC-derived motor neurons instead. With that, I've given you a preview of what our research is all about. So like already mentioned, I am part of the FIRST lab. We also post a lot on our social media. So if you're interested in following our research updates, you can always scan this code. I'd also like to thank all of our funders, our collaborators, and also the organizers for inviting us. If you have any questions, I'd be happy to answer them, but you can also contact us afterwards if you're interested in more information.
Thank you for your nice presentation. I actually have one question. So you mentioned that you collected teeth from different people from different countries even, but does that mean that you would be able in the future to, for example, use the tooth of someone to use it as a personalized to try to personalize medicines in the future?
That's a very interesting question. In theory, it is possible because, of course, it's very, there are also a lot of mechanisms, epigenetic mechanisms present. So, and we do see differences between our different patients. So it could be a very personalized approach. But of course, we are not encouraging people to donate teeth if they do not have to have them extracted for actual surgical reasons.
Yeah, indeed. There are some questions in the chat. So the first one, are there lineage-based advantage of adults vs primary teeth at the source of DPSC? And since the later will be easier to access in larger amounts for disease that occur earlier, especially.
Yes, so this is indeed a very interesting question. There are some differences if we would isolate these stem cells from the pulp of the primary teeth or the permanent teeth. They're actually also considered different cell types, so they're called sheds, but they are also from the dental pulp. What we do see is that these cells from actual children, so the primary teeth, is that they do have similar characteristics. They are, however, more immature, more proliferative, which could be interesting for certain applications. But if you want to, like with us, to differentiate our cells as far as possible and Schwann cell lineage, the adult teeth are more interesting. But this is definitely something that is currently under research a lot. And I believe there are even some tooth biobanks to which you can donate your teeth that you lose when you're a kid. Yeah, so it's definitely something that is happening a lot currently.
Okay. The next question is, are you able to share the constituents of your preservation medium?
I'm not sure if I can, but it actually is very simple. So we actually just basically left all animal and products out of it to be able to have very basal medium with just a higher amount of antibiotics and also antifungal products to really limit mainly the possibility of infections during the transport, because it is sometimes quite long transport. But if you're interested, you can definitely always contact us, and maybe then we can see for more information.
And then there are 3 more practical questions. The first one is, how did you tackle fibroblast overgrowth using the explant method?
Yes, so of course, that we have to be aware of the fact that our population that we're working with is a bit heterogeneous. For the fibroblast overgrowth, well, we do see that the majority of our cells are indeed pure TPSCs. So we check this by flux to check for the percentage of mesenchymal stem cell markers, and they do very well. So we also do not sort them further. It could be a possibility for further research. I know some labs do it as well, but we use the entire population that we isolate. When we differentiate them to Schwann cells, we also have more precise redirectment of these all cell types that are present, and we actually do not see any fibroblasts in our Schwann cell populations, and we also see that they all express Schwann cell markers, so we're not really worried a lot about possible contamination.
Okay. Did you try enzymatic isolation protocols, and what are the difference in, for example, yield, purity, dedifferentiation with the explant method?
Yes, this is indeed something that our team has done in the past. We actually have a publication about it. There are not a lot of differences in the yield of cells. If I remember correctly, by using the explant method, the resulting cells were a little less heterogeneous. And also it's a little bit more practical in use. But other than that, they are quite similar, but it has been validated.
Okay. Have you also tried to isolate from other teeth, for example, incisors?
Yeah, we have actually, definitely with the patient-derived teeth, because we accept any type of teeth that patients can give us. So there are some non-wisdom teeth that are present, and this has also successfully been done.
Okay, I think it's the last question. How do you match CMTA samples to healthy control? For example, comparing samples from persons of the same or similar age, sex, or ethnicity.
Yeah, this is indeed, like you said, this is we keep in mind the age, the gender. So we try to match this as much as possible. Of course, there's still a bit of variability in that as well. On top of that, we have some patients that are actually a bit older, that are like, the oldest one is even 6 years old. And usually people don't have their wisdom teeth anymore at that age. So we do not have age match controls for all of them, but we really try to validate a lot and to match with different healthy donors as well. And we see quite a lot of consistency in that.
All right. I see a last minute question. I know they are. Someone has put the link to your article in the. Perfect. So thank you very much for your talk, both of you. It was a very nice presentation. Thank you to the participants for attending this webinar. If you have any feedback or any information you would like to share with us, don't hesitate to complete the survey that will be available when you close this webinar. There is the next webinar will be organized in December on the 19th, and will be focusing on organoids and organ-on-chip models. I will put the registration link in the chat. So if you are interested, don't hesitate to join us during the next session. And if you would like to receive the certificate of attendance, don't hesitate to contact me or to put your email address in the chat, and I will provide the certificates in the coming weeks. Thanks a lot to everyone, and I hope to see you during the next webinars. Bye. Thank you.